have begun. First order of business, everybody. Where during that week, pick a day, 18, 20th or 22nd. Friday, Friday, all happy dance. We're doing it the 22nd. There's, we'll have to squeeze another exam in before the final in that last month. So I'll have more information. I'll work on that and figure out what we're going to do about that after the break. We'll go from there. So understand the mindset after break for many of you who have not sprung break. Is that the appropriate term? Sprung and broken. When you come back, you got to get back into the term mode because most people are on. It's almost over. I understand that, but it means that. But that's when you pile it on.
Yeah, I've noticed.
Hey, have you noticed that?
They're already doing it.
Ah, bless them. All right, so enough of that locker room talk. We're on our merry way. We are finishing viruses today. We do. And so up on the board, as you can see there in blue color, on the far side, if you can't see it, you go over, take a picture, whatever you want to do. This is the nugget with regard to cancer. And I just looked at Calvin's PowerPoint, they're saying 13%. This says 15 of all cancers caused by viruses. These are the mechanisms. These are similar when you undoubtedly all have become familiar with the breast cancer genes, BRCA1 and BRCA2, and understand that those genes are in the category of what are called tumor suppressor genes. So you have two these are the two kinds of genes that are related to cancer and viruses. So the key is the virus fusing. That's here. So that mutation basically is a viral mutation. When that occurs, when it damages the host cell in some way, shape, or form, it affects where we all have these tumor cells. They're called proto-oncogenes. They are, by definition, inactive. Okay? They are not something that their whole idea is to keep tumors from occurring. They go from inactive to active. When the viral mutations activate them, they become oncogenes. Onco means tumor. So at that point, they accelerate growth of tumors or potential tumors. Think of that, this analogy, when I came from a wonderful proof writing book called Emperor Mal, like life history of cancer. And effectively, that's like you're in the car and you're accelerating and something stop resetting. That's what I'm doing. The flip side of that is your brakes. Tumor suppressor genes. They are always active. These would suggest tumors growth occur. They inducing apoptosis, all of that. If we take those away, so they're flip side of that, always active. And what happens, they inactivate in the presence of certain viral mutations. So effectively, lose your brakes. So those brakes are there to prevent tumors. Once you lose them, increase tumors. Once the proto-oncogenes turn to oncogenes A, tumor growth. So imagine you're in a car and all of a sudden the accelerator gets stuck and you have no brakes. That's a virus. It's the easiest way to express it. And so we have now begun to categorize different types of genetic mutations in our cells secondary to viruses, and that's how they analyze the BRCA, breast cancer 1 and breast cancer 2 genes. So the different, and I'm going to spare you the discussion. We use the term tumor incorrectly. Tumor, the implication is cancer. Most tumors are benign. When abnormal growth of a normal cell is a tumor, abnormal growth of an abnormal cell is a malignant tumor, vis-a-vis cancer. Okay, that's what. So proto-oncogenes, good guys. Tumor suppression genes, good guys. Mutations, altogether bad. So, and there's different ways: viruses, chronic infections, etc. So when we look at the typical, when we get to this in urinary tract, we'll look at HPV and we'll look at those particular subtypes of HPV, and effectively those are the ones that the HPV vaccines originally Gardasil, and there are probably other ones named that was the name was marketed under employed. These are examples, and you should know these: Epstein-Barr, hepatitis B, HHV8, that's the Kaposi sarcoma, Hep B, and more famously now Hep C, liver cancer. It's a little different, both the same gene, but the liver is one of those interesting glands or organs rather that reproduces extraordinarily fast. We now do live liver transplants rather than having to get them from a deceased donor. So those cells replicate so rapidly that you're at a tipping point where a chronic viral infection like hepatitis B or C typically leads. And it's not the only thing, obviously. Chronic alcohol use predisposed to, etc. But of the viruses we have looked at, ignore this one because it's very rare. It's HPVL, which is actually a retrovirus. HPV, Epstein-Barr, that's the one for mononucleosis. That's HHV4, HHV8, which is Kaposi sarcoma. Those are all big players. Comes to, it's truly HPV predominant. So that's that. Not going through all that or how you culture them. We typically use poultry eggs when they make like flu vaccine, or I have no idea how they, perhaps it's a different mechanism in COVID era. So I'm not familiar with how that vaccine, I would be surprised if it did that. If you didn't know that. These are called cytopathic effects. Viruses don't make toxins. Viruses damage cells when they damage them. Okay, and so typically when we see evidence of it, of altered genetic material in the nucleus that has an odd stain, we call that an inclusion body. So when we're looking pathologically, we're looking at slides to determine whether or not this is cancerous, whether it's a viral-related cancer or a viral infection. We look for that. The other one is, remember we talked about RSV, respiratory syncytial virus. This is one of those nasty ways it gets cells to fuse, and they basically lose function. So in the lung, if it's particularly devastating in newborns. And finding out most recently in older people, RSV is nasty. And a lot of you will have been exposed to it and they have had it. And the healthier you are, it's not likely to be fatal unless you're very young and very old. So we're not going to do any counting or anything like that, and we're not going to do those tests. And we're almost done with this. I'm going to go over the Cowan stuff if there's any little, which brings us to the last aspect of this, which are called prions, which is short for proteinaceous infectious agents. Misfolded protein, acellular in nature. Short answer, we don't know a ton about them. But we know that they're slow, in a sense, 2 to 10 years from the time of the infection to the time of fatality. And usually, and I do not know if that is true, because when they say usually it's species to species, deer to deer, sheep to sheep. But mad-cow disease, most famously, because if you did not know, in England, it's a very much a meat eating culture, very, very much so. So that's the nature of that. You know, they do, different areas have certain animal sources they use extensively. So where India, because of the cows are sacred, is certainly not a meat eating culture. Okay, where they don't, in an area where lamb grows better, let's say Mediterranean area, North Africa, more lamb, because it's very difficult for to have cows and other things. So it's different cultures have different ones. England's very beefy, to say the least, and that. But here you have a transmission from something that ostensibly formed in cows to consuming by people and killing a lot of people, and that was really the onset of her identification. So what do we know about it? Prion diseases, if you can try, we don't know how they spread necessarily. We think it's consumption, eating them. Okay, typically we think they affect the nervous system, so it's effectively eating elements of the nervous system. I mean, so think of it as consuming brain, consuming spinal fluids. Okay. I didn't bring that one. Or we can start. Not with the chicken, but so what do we know about them? One, they're always fatal. They're always slow, so it takes a long time. And slow, just like HIV/AIDS, 2 to 10 years. Okay, and what they do is cause over time neurodegenerative changes, and they're classified as fungiform because actually the brain post-mortem has little holes in it almost, with fungiform encephalopathy. Fortunately, we can see that on MRI. Yeah, so it's not, maybe it'll make a diagnosis. I don't know if you're making treatment. That's what we know about it. Okay. How it's passed, how we get rid of it, not clear. I would not go to bed worrying about prions. Okay. What they found in England when this developed, again, okay, when we say something is banned or illegal, doesn't mean it's not done. I broke the law getting to work because I was over the speed limit. Many times we all are when we're driving. Okay, some of us more than others. My eldest was on a first name basis with the traffic court judge. Just my boys were sports car, 101. So what they did when the only way they did not know where they would trace to where the cows came from when people were dying and they identified the source. And the only thing they could do was to effectively eliminate the herd. Any members of that herd or their offspring were destroyed. Okay. Why this was happening, they thought it was something called bone meal. So when cows were slaughtered, and this is true for a lot of animals, what they would do is you have the meat that you use for meat, you'd have the hide that you use for leather, okay, and what did you have left? A whole bunch of stuff, bones, brains, nervous system, organs, etc., that kind of stuff. They would grind that up and call it bone meal and feed that back to the hive, to the herd. That's that was you're not supposed to do that. That was done in this country. A lot of other countries don't trust meat that they don't know about the source. Like for a long time, and maybe still, Japan would not buy meat from this country. They would not, because they were concerned about it. They were concerned about it in Canada, meat from that source. So understand that not everybody is sure that this has been stopped. I wouldn't go. The hamburger I had for dinner last night, I am not worried. Okay, so I don't go. Give yourself worry lines for that. That is what was done. And that was done in dog food and things like that. They put bone meal in dog food. Now we're much more with pet food, we're much more on top of things. For a long time, we made our own dog food and things like that. So some of us really think that these may all be either similar or even the same with subtle variations. It may be one big prion family that's causing all of this. So sheeps and goats, here's the mad cow, and they all typically spongiform encephalopathy tells you what it is. Deer and elk, mink, antelope, cats. This is interesting. This is where we got the idea. They were able to bring up effectively mummified specimens of cannibalistic tribes and found that they had effectively spongiform encephalopathy. The prion was named as was named one of those was named for it. The disease is called Kuru. So and I don't think they were discriminating about whose brains or whose nervous system elements they were ingesting. Probably just I'd like a little salt with that. Trying to be funny, obviously not succeeding. So we do have some inherited types which we're trying to understand. It's got a way of pronouncing called Creutzfeldt-Jakob disease. Okay, and so you can have the variant one, which they think is for how the mad cow was transmitted. Not that you have to be crazy about knowing all this stuff, or the inherited form. So there's a lot of interesting variants in this sort of assembly factor. What would I say about it? One thing that it does is it causes proteins to misfold. When proteins misfold, they're irregular. So they cause something in the brain called neurofibrillary tangles. That's one of the hallmarks of Alzheimer's disease. That's not to say that Alzheimer's is caused by this. The reality is we think about 5% of things that we categorize in the Alzheimer's family, senile dementias, probably have a source with this. Just saying. I had a student whose husband was a researcher at Pitt, who was doing his doctoral research on this particular area. Just in this, this is 10, 15 years ago. So you can see these are the holes. So here, holes develop, the nerves died, deteriorates. So we give them the name transmissible because as far as we know, they are contagious, not clear how they spread, spongiform encephalopathies. So effectively, a misfolded protein will eventually cause multiple proteins. And yes, they are more resistant than endospores in efforts to sterilization. That's why they never were, and still are not included in the definition of sterilization. So we'll see if the other PowerPoint has anything else relevant to say. This is roughly where we left off of things that I haven't covered already. This is the Cowan PowerPoint, that chapter 5. Cytopathic effects, but most of the damage from viruses is immunological. They trigger your immune system, that common cold, that flu, that COVID, that RSV. The RSV because it forms syncytia, but a lot of it is responses of your immunological system, pro-inflammatory agents making your immune system more aggressive that are there. So they talk about the inclusion bodies, which are more of evidence, and the syncytia, which are more of evidence that are there. And really, it's the accumulated damage and we learned a lot about that, particularly during the COVID era, the whole thing about what we call comorbidities. The healthier you are, the more able you are typically to survive diseases. You will see when we get to respiratory diseases and certain unusual strains of influenza, that's not always true because of the immune system. When your immune system goes crazy, boils over, you are in big trouble. You're showing you some pictures. I mean, this is the idea when it becomes latent or chronically latent, not a big deal. This is their estimate on oncogenic or cancer-causing viruses. And again, some correctly cause cancers. Others, like I'm pointing out up there, produce proteins that basically impact the oncogene and tumor suppressor gene population that are there. These are just, again, the big ones for us are a number of herpes viruses, Hep B, they should have included Hep C, and all of the different papillomaviruses. I'm not worried about this because it's exceedingly rare. This is the bacteriophage. Really not. I covered as much of that as I was interested in, and that is really the lysogenic conversion you should know about with regard to the accumulation of or the development of toxins, of things that make that particular bacteria nastier. Okay, this is all cultivating. Uh, I know they must undoubtedly still do these things today. Bird embryos, perhaps more so than anything else. This is all what it looks like. Not too interested in that. Here's the prions. So again, this is that long fibrils. These, again, neurofibrillary tangles. So if you end up, I don't know if it was presented to you in AP1. I always do a little bit of it when I do AP1 with regard to the Alzheimer's. And it's a very, very interesting disease in itself. And I have, again, my mom succumbed to that. Had a great run. My mom died when she was 93. So, I mean, it's not, I'm not complaining. My mom was driving a car when she was 86 years old. And I couldn't keep up with her. Oh, I love this story. She got her first major speeding ticket, I think she was 79. There was a place where I grew up, in the town I grew up with, called Springfield, New Jersey, in North Jersey. We used to race. It's above, if you're into golf, above Walters Royal Golf Course, which they always have the PGA, the US Open there. Famous golf course. And so when we were teenagers, we'd race up there. The cops would grab us and everything like that. My mom was taking that for visiting a friend up to where she lived in a neighboring town. And all of a sudden, and she's flying. It's a 25. She's doing at least 45, she said, maybe higher. And she's driving her Maxim. She's got a fairly sporty car. Zips over, and obviously sees the lights, immediately pulls over. And the cop, she said, she watched him inside. The cop came out storming. He figured some kid who borrowed mom and dad's car was racing it. And he comes to the window. Do you know? Oh, I'm sorry, ma'am. Do you know you were going too fast? Yes, I do, officer. She was like 49 or 25. Go, Ruth. Her motto was, life was not supposed to be spent in a car. This is what, and I drive, pardon the expression, like an old man. I drive slowly. All right, enough. We're not doing viroids. They affect plants. Okay, all of these things worldwide. Yellow fever, dengue. Again, we see that a lot. It's just about mosquitoes. All the things affect us constantly. High mortality, rabies, AIDS, Ebola. That's very fair. Very, very fair. Polio, neonatal rubella. That's what I was talking about. We'll go over it again with skin diseases, and we talk about all the neurological things. Chronic infections. Look at that. Type 1 diabetes. The current theory, and research bears it out, that this is an autoimmune disease, probably from a viral transmission early on. Multiple MS, that was that human herpes 6. We think there's a strong correlation. Cancers, Alzheimer's, obesity. I'm not as familiar with the causes. That'd be great for me. I could explain it away. My dad always told me how to do more exercises. He goes, this one, push away the food. See, I missed it. I didn't tell you that story, did I? See, on my dad's side of the family, I had two had two. I only have one male cousin left on my dad's side. Both those guys were like one was 6'4", one was 6'3". One is 6'3". Okay, skinny as rails. My dad was probably, he and his brother were probably the shortest in the family. My mom's side, the family name is Krumholtz. Okay, Holtz means woods. Normally in Austria, Poland, they were Polish areas near Krakow. Okay. Normally they had the last name, the family name is Krum. Holtz meant trees, typically like tall like trees. Not us, tree trunks. Honest to goodness. My uncle, his two boys, this is our general shape. I'm on that side of the family. Everybody, I didn't get those genes. So I got to blame them. Moving on. Down the road, okay, we're going to get to this. Antibiotics have no effect on viruses. Sometimes they'll prescribe one for an influenza or for COVID if there's a strong suspicion that someone is predisposed to pneumonia. That, even though that's not necessarily part of the thing, we'll look at the different ways that they work.
Is that what the Paxlovid is for?
I don't know.
Okay.
I will look. I will. It's a good question. I will check it out when we get to antivirals, and that comes up. That's in our next unit. This is everything from the second exam. From now on, it's third exam we're starting. Yeah, my sister took that.
Yeah, my dad did.
Yeah, and it helped her. They had no problem, and they flew back on several flights from one of their many European vacations. It was like 2 years ago. I got. She had every vaccination possible. Typical. Oh. She's the one who would refuse to go to a public bathroom. It's okay. She's smarter than I am, and she's better looking than I am. She's 5 years, 7 months, 1 day, and 1 hour older. Most importantly, I like the older part. Okay. And now we begin the new unit. So I'm going to end up using two. And this is it's entitled, as you can see here, Interactions Between Microbes and Humans. This is how they affect us. It involves how it causes damage. It involves how we transmit it. It involves aspects of epidemiology. I do not know if you will do this in lab. I'll ask Dr. Dress if he's doing it this year. I think it's fun. It's called a synthetic epidemic, and you kind of simulate a blood-borne or fluid-borne passing. It could be anything. It could be influenza. It could be an STD. It could be whatever you want. Okay, it's kind of a fun lab to do. I'll ask him. He may or may not, because I know he's got a full schedule of stuff that he does. So will be some terminology in this as well, and I think it's important because we use terminology. We have gone nuts. Every time someone says there's an epidemic or a pandemic, everybody goes crazy. All right, COVID notwithstanding. What does it mean to be a pandemic? An epidemic means disease that we could have an epidemic of the common cold. Okay, that means we have statistical information as to what would be the expected numbers of colds at this time of year. If we have a greater amount in a particular area, we call that an epidemic. If it crosses an ocean, if it goes from one continent to another, it's a pandemic. I would not worry if there was a pandemic of a new strain of the common cold that was not fatal. Don't care. So we use the terms, they're buzzwords. So as you guys get farther along in your careers, with families, with everything else, don't be fooled by things. They use a lot of buzzwords to sell product. They use a lot of buzzwords to make you buy medicine, to make you do things. Learned about this in COVID. People have a tendency to overreact and particularly when there's a possibility of profit, fortunately. So we are formed and react to a lot by our environment, as we all know. The microbiome are all these microbial forms that we have talked about so far that are part of us. They're either on us, and we're totally covered in bacteria. We're totally covered in yeast or fungi. Totally. We have viruses every time we inhale. So either if they're not on us, they're close to us. Okay. And really what we do, and part of good health, is being able to interact with them on a regular basis. If they're good guys, we get to know them. They cover up areas where bad guys can be. They gobble up nutrients that bad guys could use. By the same token, if they're bad guys, we become familiar with them early on and can attack them. Okay, that's this whole idea, and I mentioned it in passing before, the hygiene hypothesis. This idea of being in a scrupulously sterile environment. The trouble is your immune system has to constantly be challenged. It's constantly sampling the environment. I mean, I don't know what parenting is like now. I had my kids, I didn't have to worry about. They wanted to be outside and go crazy. Okay. I don't know if they're being become overprotective because of COVID or because of other things, and maybe you're seeing it. But our moms and dads threw us out of the house. We got home from school and said, "I don't want to see you back here at 6 o'clock." Basically, out there, play, come in, get dirty, yell you got dirty, and they'd secretly be happy about it. I told you about the chicken pox and measles parties. They wanted you to get that stuff. If it doesn't kill you, it makes you stronger. If you want to think of it, maybe that's a poor way to express it. But that's what the hygiene hypothesis was wrong about. That lack of exposure to what every other kid has is a problem. I can't say that enough. So they colonize us long term and they don't cause disease. If it's potentially a disease-causing organism and it gets past our defenses, many of you have not had, for you guys in AP2, have you had the immunology nugget yet? Have you started that yet? Probably not. Generally started after break. And immunology is all about you've got three levels of your defense. The ones that makes antibodies is your third level. By and large, your defenses are very, very strong on the outside. 2 levels. But if it gets past that and gets to our interior defenses, that's when we have to rely on this third line that it either makes antibodies or makes cells to combat it. So and if it effectively it's a disease when it does some form of disruption, if you want to think of it that way. An infectious disease by definition. So there's lots of diseases that aren't infectious by nature. Okay. Atherosclerotic heart disease. Okay. You're overweight, you don't exercise, maybe you have type 2 diabetes over time. You have a disease that is potentially and frequently fatal. But the reality is it's not an infectious disease. Type 1, however, we now believe it is, has a very strong autoimmune and perhaps viral component. So a pathological state caused directly, whether it's that cold, whether it's that stomach upset, whether it is, COVID itself or influenza. So this started in 2007 to determine how they differ, various diseases. We have the availability now of genomic sequencing. There's so a lot. We're even doing that here. So, I mean, you would not be here if you were in those courses. But if you're one of our bio-related majors, they're doing PCR. They're doing typing. They're doing bands to be able to identify genetic sequences that are there. And it's commonplace. It's not just, we're not anything special. We probably do it to a lesser degree than big, larger universities that do this type of stuff. So it is. So typically, these are the results. And I've seen the number here. when they talk about a gene, basically it encodes a protein or perhaps even a sequence of proteins. We have about 21,000 that are there. Okay? Again, only about maybe 3% of our genetic material really is responsible for this. Millions of protein encoding genes are found by the accumulation of the microbes. So, and we even found them in areas where we didn't think they were. In areas we thought, when we talk about sterile tissue, if you talk about your abdominal peritoneal cavity, okay, if someone had, that's a sterile environment as far as we know. If you rupture an appendix, it's no longer sterile because the appendix communicates with the gastrointestinal tract, and that's by no means sterile. It's filled with bacteria. 100 million viruses, we didn't think they were that abundant in the gastrointestinal tract. Okay, and to a certain extent, we have a lot of these things. We all, to be sure, we all have C. diff sitting inside of us. We all have Staph aureus on our skin. We all have Staph aureus on our nose. If you're in healthcare, at least twice a year, you're cultivating MRSA in your nose if you work in a hospital. Doesn't matter whether you wear the mask or not. It's just, eventually, it's going to get in there. Okay, and there's a lot of overall influences. So take a look. Sites previously known, anywhere on your skin, anywhere inside your mucous membranes, any of the linings, mouth, nose, anogenital region, you figure it. Okay, GI, outer portion urethra, external genitalia, vaginally, external ear, eye, etc. Lungs, you're inhaling microorganisms. Some of them are going to get down here. You have, if you did respiratory in AP2, you probably didn't do that either, did you? Okay.
Or in cardiovascular.
You're in cardiovascular, and that's about where I would anticipate you would be. I know what I usually do in that unit. That's my second unit to complete in AP2. And because I do I typically do blood vessels and immune for my third test, and then everything else, respiratory, digestive, to the final. Let's see. What I was saying is it shouldn't surprise you. You're going to learn that we literally daily, we're destroying millions of macrophages that are like little sweepers in your lung, gobbling up stuff that got there that shouldn't be there. It's like you had one of the, I always use it has like one of those neat Roomba things, My stepson and his wife, and they got a beautiful home, and have a lot of those hardwood, and that Roomba, all of a sudden someone runs into you. What? It's a Roomba. Bladder and urine, I can account for that. I can tell you about that. Breast and breast milk was surprising. Amniotic fluid in and around the fetus itself. Okay. DNA has been detected even in there, has somehow been destroyed, and some of the DNA has escaped. It's shocking. And all of these things, and understand we have a lot of places where we inhabit. Maybe the vast majority of us are like us here in a relatively speaking temperate climate. We're not necessarily around the equator. We're north of, Japan and Korea are basically the same kind of climate that we have here. Large populations. India, the subcontinent particularly, is much more equatorial in nature. For instance, China probably more similar to ours than not. Okay. So we look at a type of things like that. So they can be displaced. There's only so many sites that they can attach to. Okay. And this is the advantage for typical microbiome. And the term I always use. So forgive me if I go back to it's called normal flora. Then we used to think they were plants. So we use the term flora. So now it's normal flora or normal microflora. Okay, so it's beneficial or at worst commensal. So we have different kinds of interactions between different hosts, between a host and a microbe. You can have what's called, and they'll probably define these, mutualism and commensalism, which I can't distinguish. Okay, mutualism means both parties benefit. Okay, commensalism is one isn't harmed and the other one benefits. To me, no harm, no foul. Nobody's being damaged. A parasite, by definition, and parasites in medicine, we talk about eukaryotic pathogens like fungi, like protozoans. So you got to understand the context of these questions if you're asked them. But a parasite, like a bacteria or virus or one of those other things, is thriving while damage is being done to the host. That's kind of the way. So it's not those normal ones are fine. Things that weaken it. And these are COVID days, they coined the term comorbidities. You know, we called it immunocompromised, compromised host, whatever you want to call it. Very young, very old, makes sense. Genetic defects in immunity, or ones that have been acquired, like AIDS, previous operation. When you have transplants, inevitably we have to use immunosuppressive medications. I was on, and I've stopped taking. I'm going to see how it works out, an immunosuppressive medication. I've gotten to the point where I think by controlling diet, I can do better with it. Diet and, God forbid, exercise. I know. It's okay. I got up and chased the dog out. And went up and down the stairs to let him back in. I do a lot. Oh, God, my son has three. His apartment's on the third floor. And I'm carrying up tires and tools. So, immunosuppressant agent or immunosuppressed for any other reasons, any underlying disease, variety of those drugs, stressors, pregnancy, other infections. This is interesting. When we begin to understand that these are areas that potentially problematic. When we get into a bit into the urinary tract infections, you're going to see there's a lot of concern that babies can be significantly harmed during the birth process, particularly with the first case of genital herpes. And they probably don't have the images anymore in the PowerPoint. They are scary. Okay. So a lot of the normal microbes are picked up through vaginal birth process. Lactobacillus is really important. That's a big player vaginally. It's an extraordinarily important over time. It's the single most important one for females during the menstrual year or childbearing years for protection by far and away. So as you can see, 600 species of bacteria and sugars a baby cannot digest but are digested by the bacteria. If you didn't know it, effectively babies are all lactose. We all start off lactose intolerant. Okay, that's the reason why we don't use milk-based formula unless you're putting additives or something like that. And there's a whole zillion. And inevitably, most of us have kids that have to be on some kind of special formula. I don't know. My kids were on soy formula, and none of them, again, they're all adopted. Another one had to be on one that was pre-digested because he had such trouble with his gut. Thankfully grew out of it. So they're going all of this different. You can look at that as well as I can read it to you. I'm not going to go over the questions, et cetera, et cetera. This is what we're talking about. So it's not all about the pathogens, but we focus on, in this chapter, a lot about how they do damage, how they come to us, how we spread them. Intuitively, you get a certain amount of prevention. So by definition, pathogen, disease causing, a microbe whose relationship with its host is parasitic, it thrives, harms you, and results in infection and disease. We talk about this a lot. We're going to talk about things called virulence factors. That's its nastiness index. How quickly it affects you, how rapidly you get sick, how dangerous it is to you, how easily it's passed on, how many it requires. There's something called infecting dose. Some diseases you've got to encounter a ton of them, some not so many. So the pathogenicity is caused a lot by virulence factors. One of them is a true pathogen capable of causing disease in healthy persons with normal immune defenses. Okay. Pick something we don't see, the plague. That would be right in there. Okay. Smallpox, as far as we know, eradicated. Be right in there. And there are many of them. Theoretically, you work with one in the lab. Staphylococcus aureus is by definition a true pathogen. We realize that that for us, there has changed. The laboratory strains that they send, that you work with in the lab, are effectively not dangerous as long as they are handled properly. And I've always told students, that, hey, if you've got some underlying condition, to always let me know privately. If you would rather not work with a certain, if you're like, you do antibiotic testing yet? Little distant, okay. I mean, you work with penicillin. If you're allergic to penicillin, don't touch it. Somebody else will do it. I'll do it. Dr. Dress will do it. A lot of problems are opportunistic pathogens. If you're going to work in a hospital, as many of you most likely will, you encounter these more than anything else. They affect people with compromised hosts, bad defenses, and it goes someplace where it shouldn't. You heard all about MRSA. We're going to hear a whole lot about MRSA, methicillin-resistant Staphylococcus epidermidis. Staphylococcus epidermidis is on all of our skin all of the time. Okay, these are the virulence idea. Relative severity, and it correlates to its degree of pathogenicity. So, how easy it is to establish itself, how much damage. All of these are virulence factors. Anything, any characteristic or structure that contributes to its ability to establish, etc., etc. We'll get to it. We'll stop here. More on that next time. Again, nothing Friday. If I'm feeling up to it, I'll record something for Friday.
Don't worry about that.
You don't just, oh, thanks.
I won't be here.
Enjoy your vacation.
Oh, yeah, hey. I will make an effort. Thank you, everybody. See you all Wednesday.